So yeah, let's start with some definitions. What is a cryptic pocket? So you have a nice example here on the left. It is like TEM1 beta-lactamase, and you have a hollow structure of beta-lactamase with a ligand bound to an allosteric cryptic site, and you have a corresponding apo surface structure of the same protein. And as you can notice, in the case of the apo structure, the same surface is completely flat. This is like the definition of a cryptic pocket. Basically, is a pocket that is like only detectable in its ligand bound state, and is not easily detectable in its apo state. A conformational change is required in order to expose these kind of cavities. And we can have different mechanisms that actually underpin the opening of these kind of cryptic sites. We can think about a mechanism like conformational selections, in which we have like an equilibrium between open and closed states of the pockets. Or we can also have, like, of course, ligand-induced mechanism, in which like the ligand is actually inducing the opening of the pocket, and sometimes also a mix of the two. So now that we know what a cryptic pocket is, we might be wondering, do they actually have a functional role? And this is a very nice example from the Bowman's lab, in which here we still have like the same protein as before. It is TEM1 beta-lactamase. And you can see in white, you have a closed configuration of a pocket, which is located underneath this omega loop next to the active site of the enzyme, and now open conformation of the same pocket in green here. And what they did here was basically introducing some mutations in order to switch the populations between open and closed conformations of this pocket. And they also related like these mutations and these populations to different catalytic activity of the enzyme, meaning that like the opening and closing of this cryptic pocket can actually be linked to different catalytic activity of the same enzyme. So meaning, yes, at least for this case, they do have a functional role. So now let's ask ourselves, why are we interested in cryptic pockets when it comes to drug discovery? Well, we can start with some numbers. So we have more or less 20,000 genes in the human genome. And then if we focus only on the genes that are like disease-modifying genes, and they encode, of course, for protein that, if targeted, can actually have an effect on some like pathological pathway, we are left with 3,000 possible targets. And if we intersect this set with the set of druggable proteins, then we only have between 600 to 1,500 possible drug targets. Currently, I think we are targeting 400 different targets. We're kind of like close to a theoretical limit. But like this druggable genome definition is quite old and is based on sequence of known drug targets. So we can probably like play around with this definition in order to expand a bit this set. Moreover, if we look at like how much we actually know about the human proteome, it is like a very nice initiative from 2014. So here we assign basically each protein in the human proteome to a target developmental level. And we can see that only 10%, which is the clinical and the chem, are associated to possible binders. I mean, are proteins that we know can bind something, and like 55% instead are not like such. I mean, we don't know much about those proteins, and we know basically nothing about the rest of the 35%. So basically, this is like indicative of like we have still a lot of room in order to try to expand this kind of definition in order to actually enhance like number of targets we can actually use in drug discovery. And cryptic pockets is actually available strategy in order to expand this kind of drug target set. So how many cryptic pockets do we know of? Well, if we look at the PDB, based on like Sally's lab definition, actually, we have like 100 pairs of apo-holo structures of cryptic pockets, and these like apo-holo pairs were used in order to train a super vector machine to actually predict novel cryptic sites in novel targets. So the Sally's group actually in 2016 developed CryptoSite, and is like fairly accurate in locating like cryptic sites on protein targets. And then we can see, based on the estimate, that like at least 30% of disease-associated human proteins should harbor like cryptic pockets. More recently, Bowman's group actually developed a graph neural network-based approach, and they came to similar conclusions. So once they ran like the training model on a set of 10,000 different proteins, they also found that like at least like 30% should harbor cryptic pockets, meaning that like, yes, as for now, we have a limited amount of examples in the PDB, but we expect to find many more in the coming years. So how do we actually look for cryptic pockets? We have like mainly two different approaches. One is an experimental approach that boils down mostly to crystal soaking, co-crystallization, NMR experiments, and tethering. And we have a very nice example of this in KRAS. So we know that the RAS gene family is associated to a different numbers of tumors in humans, and that makes KRAS an extremely interesting therapeutic target. People tried for 30 years to actually develop ligand that could bind to the active site of KRAS. But since KRAS has a picomolar affinity for the native substrate, GTP, it was extremely hard to actually develop something binding to the active site, actually impossible. And only in 2013, Shokat and colleagues actually like exploited a mutated cysteine in the active site in order to covalently bind a ligand to the region. But once they actually resolved the structure, the ligand they covalently bound to the cysteine was not targeting the active site directly, but as you can see, it was targeting a completely unknown new allosteric cryptic pocket on the site. So yes, experimental approaches can be quite useful, and they are in detecting cryptic pockets, but they often like depend on serendipitous discovery of this kind of bending pocket. So we have, of course, like a different approach, which is what we're taking in Gervasio's group. And this is like based on MD simulations mostly. So as you know, MD simulation basically evolved like along like time a system of atoms integrating Newton's equation of motions. And we have examples, is in particular coming from the Dishon group, of like MD simulations being effective in actually detecting the formation of cryptic pockets and also being able to capture the binding of a ligand to a cryptic pocket. So as you can see in the movie here, we start from an apo structure of this target, EL2, and we actually, maybe I can play it again. And we actually sample the opening of a cryptic pocket along the simulations, and then later on the ligand can bind, resembling closely the crystallographic pulse. So this is quite nice. Still, MD simulations have some limits. The major limitation is the time scale problem. So we are interested in conformational rearrangements when it comes to cryptic pockets that expose the cavity. The issue is that we can only sample a limited time window with like MD simulations. So, and this time window corresponds to conformational rearrangements that can be described with sidechain motions and loop movements. So something like fairly easy. And we're talking about something that can happen in the nanosecond to like few microsecond time scale. When it actually comes to more complex conformational rearrangements associated to cryptic pocket opening, then we have a problem because these rearrangements come with a higher free energy cost. And it means that like we would need like much longer simulation time in order to sample the opening of these pockets. And this is something that we cannot really do with like current methodologies if we only rely on unbiased MD. So if we think about like the two like pocket configuration, open and close, as like two different accessible states in the free energy landscape of the system, if we simulate this system with unbiased MD, we're going to get stuck in an energy basin, and we're never going to actually like overcome the energetic barrier in order to sample the open pocket configuration. We have strategies to tackle this issue, and we can actually describe this in 2 different strategies. We have brute force, let's say, strategies. For example, with Anton 3, these are a purpose-built computers to run MD simulations. And with this kind of hardware, we can actually simulate 100 microseconds per day in a fairly big system. Still, we only have 1 Anton 3 computer on this planet, and not everyone can access it. So it would be quite nice to actually have different strategies. And this comes in the form of a GPU's acceleration plus some elegant, let's say, algorithm in order to speed up the sampling of a free energy landscape of a system. This is typically done through multiple replicas approaches, or Markov state models approaches, or collective variable-based metadynamics, for example. So in principle, what we're doing here with these non-sampling approaches is we're filling up the free energy landscape in order to help the system overcoming the energetic barrier and actually sample both conformation, in this case, open and closed conformation of cryptic pocket. So we can maybe see a practical example of this. If we start again from TEM1 beta-lactamase, here in white, you have an apo configuration of the protein, and in pink, a holo configuration of a protein from which I remove the ligand for clarity. And you can see you need a conformation rearrangement, this helix has to move away in order to expose a much deeper cavity. So if we start from unbiased simulations, we run a 1 microsecond unbiased simulation. This is the volume profile of this region of space along the simulation, right? And if we look at the violin plot now, this corresponds to an open configuration. So I can start from the holo configuration, remove the ligand, simulate the open configuration of a pocket in order to control how the volume fluctuates along an open configuration of a pocket. And this is what I would like to get starting from the apo configuration as well. But instead, along an MD unbiased simulation, I'm not really getting close to an open state. I can do something better than this. I can actually also include, for example, benzene molecules or any organic molecule in your simulation box in order to force the sampling of these cryptic pockets. And you can see that if we do mixed solvent simulations, it's still unbiased, but we add in benzene molecules, for example, we do recover some sort of open configurations of the pocket in terms of volume, but still not extremely robust. So some people in the group came up with the idea of maybe try to enhance the sampling through a Hamiltonian replica exchange, an Hamiltonian replica exchange scheme. So what we're doing here with SWISH is basically we have multiple copies of the system, and we start parallel simulations of the system under investigation. And then we exchange structures coming from these different simulations at given time steps. Moreover, we're also tweaking water parameters in order to force the water to be more ligand-like, meaning that in practice, we're just scaling the non-bonded interaction between water molecules and apolar atoms, deepening the non-bonded potential well, and effectively making the water to have a higher affinity for protein atoms. So in this way, basically, a higher lambda values, the very ligand-like water should sample open configurations of a pocket. And if we also add benzene, for example, in the simulation box, we can exchange back at lower lambda values and stabilize the open conformation of a pocket. So now if we look at the results from a SWISH simulations for TEM1 beta-lactamase, we can still we get a higher median value of a volume for the pocket with respect to the previous mixed solvent simulation, for example. We tested this system, scheme also in other proteins, and it turns out to be quite effective in sampling the opening of cryptic pockets. So we asked ourselves if we could actually use SWISH to predict novel cryptic pockets. So we started from a SARS-CoV-2 protein, this is NSP1, and we started from a crystal structure. And if you look at the crystal structure, we can't really identify anything that looks like a proper pocket or a binding pocket, but we can still identify some groove-like regions or pocket-like regions. So when we moved to our SWISH approach, we basically started from this like crystal, and we simulated the protein with SWISH and by SMD and mixed solvent. And we looked for the opening of possible pockets along the simulation. We actually got quite interested in pocket 1 because it was like close to a biological relevant site in the protein. So we proceed to some virtual screening, and we came up with 60 ligands that were 60 fragments, sorry, that were supposed to bind to either one of the identified cryptic pockets. Then we moved to some experimental validation. So our actual collaborators managed to crystallize 5 of these fragments with within like one of the predicted pocket. We move on to look at homologous protein of NSP1. And we basically saw that like the residues lying in pocket 1 were like quite conserved across different coronavirus genera. So in principle, if we develop something like that binds to that cryptic pocket that we identify with SWISH, we should also be able to target like different COVID genera. Still, we were wondering if we could actually improve SWISH. And we came across this nice paper from Invernizzi et al., in which they use a single simulation to sample a multi-canonical ensemble. So here, instead of like sampling the canonical ensemble within one simulation, you're sampling a multi-canonical ensemble that corresponds to a different range of temperatures and pressures, for example. So this is like the example from the original paper. It is like your CV space, your volume and energy. And if you bias this CV space in a smart way, let's say, then you can actually sample the corresponding pressure values and temperature values across a range that you select at the beginning. So we can see here that like, we can like actually sample this CV space, and that corresponds to nice folding events in a Chignorin, for example, folding simulation. So we thought, okay, can we couple this with SWISH? So we basically introduced like this OPES multi-thermal approach to each of like our like SWISH replicas. So now each replica is not only sampling a different ensemble given by the scaling factor lambda, but it's also sampling an expanded ensemble that corresponds to a range of different temperatures that we usually select between 300 and 350 Kelvin. This means that like our like approach now, it should also be able to help the system overcoming like kinetic bottlenecks in the energy landscape. So we tested this again on TEM1 beta lactamase, and we checked like the performance of SWISH expanded with respect to the other methods. And like, as you can see here, SWISH expanded like can reliably actually sample like open conformation of a pocket. And it does so in a much more efficient way. So the median volume of like across the SWISH-X simulations is actually close to the open configuration of a pocket. If we look across the different lambda scaling factors between SWISH and SWISH expanded, we can see the SWISH expanded is actually better across all the lambda factors with respect to SWISH in sampling this open configuration. And also, if we focus on the first 250 nanoseconds of simulation, each replica is 500 nanoseconds in total, but if we focus only on the first 250 nanoseconds, we can see that we already get, with SWISH expanded, good sampling of a pocket, meaning that we can do this like in a much quicker way with respect to SWISH. So that was like quite promising, and we decided to move to more, let's say, interesting targets. So, and this like brought us to protein-protein interactions. Basically, as we probably all know, protein-protein interactions are extremely valuable in terms of like potential drug targets. Still, regardless of their despite their potential, it's quite difficult to target this with small molecules because the interaction surface usually is like quite flat and featureless. So like small molecules is not really extremely easy to get like a small molecule to bind at the interface. But still, I mean, we can think of some like sort of like rearrangements when like the two partner protein come to bind that can expose, for example, cryptic pockets. So we were interested in seeing if like our methods can actually detect cryptic pockets of protein-protein interfaces. So what we did was basically coming up with like this strategy. We selected 4 different protein-protein interaction interfaces for which we basically know everything. We have a hollow structure and an apo structure. And then we started from the apo structure and we simulated this protein target with like 4 different methods: unbiased MD, mixed solvent MD, SWISH and SWISH-expanded. And in parallel, we also started from the holo configuration of the same protein. We remove a ligand and we sample the open configuration of the pocket with unbiased MD. Then we basically project the different conformation into TSNE space in order to try to detect different open and closed conformation that we sampled along the different trajectories. So these are the results for BC-ELIXIR. Here again, in white, you have the apo configuration of a pocket, and in purple, instead, like the holo configuration of a pocket. And you can see that, like, in order for the white structure to expose a cavity, you need like this helix to move away and partially unfold. And you also need like this phenylalanine to flip in order to expose a deeper cavity. If we look at like how the different methods perform on this specific system, then we notice that like all the SWISH-expander is actually able to sample like open configurations of a pocket. Mixed solvent doesn't really manage to do so, and SWISH only partially sample open configurations of a pocket. So here we're starting again from like a closed configuration, and only with SWISH-expander we managed to like recover the open configuration of a pocket. So if we look more closely at the structures that we actually sample along the simulation, so here the isocounter basically delimits the TSNE space that describes open configurations of a pocket. And these different clusters are instead like different pocket configuration coming from a SWISH-expanded simulation of BC-ELIXIR. So here in purple, you have the hollow structure, and here in gray, instead, you have a representative structure of one of these clusters. And we can see that like for some clusters that are close to the isocontour or like overlapping partially, we can actually sample very similar configurations from the crystal structure. So if this line gray is like one structure coming from a SWISH-expanded simulation, and now if we activate the surface representation, we can really appreciate how close we get to the initial crystal pose. So this is like further like highlighted by coloring these cluster maps by volume values, and you can see that like different like simulation methods actually fail in capturing open configurations. So red means open basically and blue closed. Besides SWISH-expanded and SWISH, that SWISH-expanded like in a much like more robust way, and SWISH can also like somehow like sample open-like configurations of the pocket. So we apply like the same method to the other like 3 systems, EL2, MDM2, and HPV. And we can see basically from here that like SWISH-expanded is always like, and SWISH in these cases, is always able actually to start from an apo configuration of a pocket and recover, from a closed configuration of a pocket and recover the open configuration of like the pocket. Meaning that like we can apply this like approach to probably like different systems, and we're confident we can actually get a good sampling of the opening of these cryptic pockets. So I think with this done, and I would like to thank my group, like all the collaborators involved in the different projects I presented, especially Joseph, Frank, Carolina, and Eleanor. And thank you for your attention.
Yeah, thank you very much. Really very, very interesting. So do you have some questions that people put in the Q&A? I don't see any. Okay, maybe I can ask you a question. So how do you define really a cryptic pocket? In other words, you know, from the examples you give, you are, you know, it's let's say the apo conformation, which is, you know, one of the states of the protein, which is, you know, normally, you know, you have it in a crystal structure, but you know that it's functionally important. in general, you know, how, if you didn't know what is functionally important, how would you define a cryptic pocket, you know, for a drug to bind?
Yeah, I mean, usually the approach is like just comparing two different structures. So if you have an apo structure in which you can't really detect anything, but like resemble a pocket, and then you have a corresponding hollow structure in which instead you have a clear defined pocket, then you can call that site cryptic. But yeah, it's not an extremely strict definition. It's kind of like a loose definition, but it's like based on the comparison between apo and hollow structure usually.
So you basically compute, you know, the shape of the surface that you that changes, and find a place which is, you know, kind of enclosed and could bind the drug. On your, what do you characterize this? You know,
yeah,
this...
you can do this in different ways. Usually, like you can just apply a pocket detection algorithm to like the same ensemble of structures, and you can see actually like if you have like big differences between apo and like all of course structures, then you can actually say, okay, it is like a pocket, but like it's only present when something is bound to it. Instead of like, for example, if you think about active sites of like common enzymes, instead, I mean, you can always detect even like if a ligand free. So that's the major difference.
And in your procedure, do you weigh in, I mean, do you focus on a particular region of the protein, or you let the entire system relax and explore?
It is like the good thing about SWISH and SWISH-Expanded. These are like completely agnostic approaches. So we don't actually need to specifically target a specific region of a protein. We can just simulate the whole system and then post-process the trajectories and look for potential cryptic pockets. So no, we're not actually biasing specifically some regions, and we just like apply this to the whole system. Then like we can also think maybe like in protein-protein interaction cases, right? If you're most interested in the at the interface region, then maybe you can add like some specific bias to that region. So in principle, we can also target this. But the problem with cryptic pockets is that like you usually don't really know much about what to expect. So you start from an apo structure and you don't really, it's really hard for you to predict where you expect something to appear. So that's why like CV-based methods usually is not like extremely easy to pursue.
Okay. Can I ask another question? And in terms of protein-protein interfaces, you know, how do you prevent the dissociation of the protein?
Oh yeah, that's extremely important. And what we do actually, since we're like changing a lot, like the range of temperatures and also like scaling the interactions, I mean, what we need to do is basically add like a CMAP upper wall to our simulations. So basically, we usually run an unbiased simulations, like which is a control simulation. We look at the equilibrium distribution of like different contacts, and then we apply the CMAP, which is like not too strict, so it allows for some movements of the protein, and we apply that to prevent major unfolding, but still to allow like enough like flexibility in order for the pocket to actually open up.
So the last question would be, can you estimate the free energies of the differences in free energies of, or the free energies of the pocket, basically how populated they are and whether this matters?
In principle, yes, we should be able to do that, but that's something that we were trying to do, but it's very complicated. So once you have identified a potential cryptic site, okay, you can think of like maybe training some like more refined CVs in order to actually like force the transition between like open and closed states. Enough times for you to have statistics and compute actual like difference in free energies. We try to do that with ELIXIR, for example. We train a deep LDA CV, starting from a closed configuration and open configurations, but still we actually failed in converging with free energy because these things are not extremely easy to converge. Also because, like these might be transitions, like excited states. So also the solution plays an important role, because if like the opening is mostly governed by, for example, induced fit effects, like if you're simulating this in water, it just might not be a minimum in your free energy surface. But the moment you add like the right ligand, then the open configuration might switch to a minimum new free energy. So it really depends.
Do you believe in the induced fit? How, you know?
Shoshana, we have a question from the audience.
Oh, okay. I didn't see anything. Sorry. Thank you. I'm sorry. Okay. Question of the audience. Besides pair of apo and holo structures of a given pocket, do you also consider other apo and holo conformations, or did you always focus strictly on a certain pair?
Yes, I mean, like for the simulation, usually we just like decide on a pair, but like before starting the simulation, we actually look at the overall ensemble of apo and holo structures, because it can be that like maybe you do find examples of one apo structures and a holo structures, but like maybe if you look at the larger ensemble of apo structures, you might find something that actually like resemble an open configuration. So yeah, we need to take that into account. We actually look at the ensemble of structures that are available for both the apo and holo state.
Yeah. So now there are efforts, you know, by the PDBE, and that was one aspect of what we did in activity 2, is to chart the ensemble of conformations of native proteins, and then, you know, see in a way whether you can, you know, you can see something already in these ensembles or not. I mean, this is something that Sally did a number of years ago, but now you have many more examples.
Yeah, we're also actually working on something in this
Okay. Thank you very much. That was very interesting, very useful.